Hello and welcome, Central Texas Mycological Society fans. We're here for our Thursday night monthly lecture. Tonight we're joined by Daniel Reyes of Myco Alliance and one of the Central Texas Mycological Society co-founders. And so yeah, if he just wants to kind of take it away and get us going and talk about Mycoremediation tonight.
Cool. You want me to share screen now?
If you want to jump into it, or you want to introduce yourself a little bit.
Let's do that.
Maybe one or the other.
Sounds good. What's up, everybody? Looking at the participants in this session right now, I'm recognizing some names. It's really nice to see some of y'all's faces. But anyways, I'm really excited to talk to y'all tonight. My name is Daniel Reyes, as like Philip said, and like y'all probably already know. It's been kind of a crazy journey getting to this point. Some of y'all know me from Austin, from the research station. Some of y'all know me from other places. But just to give you a little bit of background in case you don't know anything about me, I am a hydrogeologist by training from the University of Texas at Austin. And I found fungi and micromediation through my work as an environmental consultant in the oil and gas industry. I won't go into all the details. If y'all have any questions about that later, I'm happy to talk more about that. But just to kind of summarize, my journey has taken me down the road of community citizen science, right? With the research station, we put together this really awesome space that we shared together with Ecology Action over at the Circle Acres Nature Preserve. And we ran that for 5 years and then slowly transitioned it over to the Mycological Society. And then nowadays there's a group dedicated to that space known as the Mycological Research Station. So if y'all know Carter and Perry and Sam, I just want to give a quick shout out to those people because they have been fundamental in keeping that project going. Also want to give a big shout out to Hi-Fi Mycology. The guys over there have been providing us with mushroom blogs for many years. And ultimately to all of you that ever came out to volunteer, to take a class, take a workshop. Honestly, it wouldn't have been possible to do everything we did there without your involvement. So thank you so much. And so I transitioned from Austin down to Mexico in 2019. I came down here in October, down to a city called Mérida in the Yucatan Peninsula. I came down here initially to teach a workshop. I was just here to, teach a 3-day workshop. And many of y'all have taken my workshop, so you know what that's about, a lecture, then followed by a hands-on activity. And initially I was gonna be here for a couple of weeks and ended up staying for 2 months. And then within those 2 months, I met a really interesting person. She is a professor at the largest university in Latin America. It's called the Universidad Nacional Autónoma de México, UNAM. And it's a really big university, and they have a campus here in the peninsula focused on environmental chemistry, environmental pollution. And this particular researcher focuses on heavy metals in soil and water. And she studies karst hydrogeology, which is my background. And so I met her. I talked to her about my background. I talked to her about what I've been doing with mushrooms. Talked to her about what I'm trying to do with micromediation. And then within an hour, she was just like, why don't you come do this here? I will fund you. We'll put together a proposal and we'll get to work. And so I ended up going back to Austin in January of 2020, right? You know, before the pandemic and all this COVID stuff happened. And some of y'all saw me mid-January, early February, giving my last workshop at the research station. And then shortly after that, I came down here. And initially I was working at the lab. I spent maybe like 15 days before the pandemic really hit. And then they sent us all home. And then, so pretty much since March of last year, the middle of the pandemic, I started a lab here in my house. My whole living room is set up to be a nice little research lab. If y'all ever visited my lab in Austin, I just have to say that this is an improvement from that. It's still not quite what I want, but it's definitely an improvement. And so I'll just cut straight to the chase. I've been doing micromediation research for the past year and a half now, and I'm really excited to cover some basics with y'all. Obviously, we're going to talk about wood degradation. We're going to talk about some basic cultivation, but we're really not going to go down that route too intensely. But I really just want to paint a big picture of what micromediation is looking like nowadays. And more than anything, I want to convey a mindset that we need to have when we approach this field of micromediation, which is becoming more and more popular. It's becoming more and more in demand. But I do feel like there's some things that we need to address, some tools, some techniques. But ultimately, it's our approach, right? It's how we relate to fungi, how we relate to these environmental problems, how we relate to each other that is going to make a difference. Right? Fungi can do so much, but it's really up to us and how we approach these problems that's going to make it successful or not successful. So, with all that being said, I want to share some slides with y'all. Some of y'all that have visited or that have come to classes and workshops before have probably seen some of these slides, so don't worry too much about it. But yeah, so let's just get down to it. We're talking about fungal wood decay processes as a basis for micromediation, right? I'm not going to go into all the details of why or how fungi break down wood at the chemical level, but I do want to do an overview of what happens, right? But just to start out, like, what are we talking about? Why do we need fungi? Like, what are we facing that is so dire that we need fungi to step in and save the day? Well, we live in a modern world. We live in a modern society full of pollutants, full of industries that are just, straight up fucking everything up, right? And if we don't do something about it, it's just going to stay like this because these are pollutants, these are constituents, these are compounds, these are chemicals. Whatever you want to categorize them, these are things that are going to stay in our environment for a really long time, and maybe eventually they'll break down because the planet is really old and it's got this really nice way of cleaning itself up. But in the meantime, we're going to be suffering because of it, right? Our activities we're going to be facing some really serious consequences because of our activities. And I'd like to bring this picture up, which I'm sure many of y'all have seen, or at least have seen a version of this, because it's not like we've got some foreign invaders, coming into our planet and doing this to us. Like, we're doing this to ourselves, right? We have been doing this to ourselves for quite a while, and we're getting worse and worse at it, or maybe better and better at it, however you want to look at it. But I just want to make it very, very clear to you that we ourselves are doing this, right? And so it's also up to us to find solutions to these problems, right? Like I said before, I came across fungi while responding to emergency events within the oil and gas industry. I was getting called up to appear at spill sites within 24 hours, full PPE gear, face masks, respirators, the full-on outfit. And my main responsibility was to come up with a remediation strategy, right? After we determined how bad of a spill it was, we were tasked with coming up with a solution for it. And just as I was doing that work, I became very disillusioned, but also, very encouraged to find alternatives to what I was seeing. The main thing that I had a problem with was that, our regulatory agencies can determine whether a site is clean, if we meet certain limits or certain concentrations, and then we're good to go in terms of the regulatory stuff, the legislative stuff, and ultimately our work is done once we meet those limits. However, you look around, I started to look around after our work was done, and I realized that we weren't really cleaning this stuff up, right? We weren't leaving it in a condition for that place to thrive and continue to provide a healthy environment or healthy conditions for other things to come in, right? We were just doing the bare minimum to get this stuff out of the way. So that's a little bit of the background, why it's important for us to work with fungi. But ultimately, and this kind of goes straight into the mindset of what I was telling you about in the intro, I would like to maybe see the day in which we don't have to work with fungi, right, to do this stuff. I would love for my job to not exist. I don't want to clean the planet with fungi. None of us should have to do this. That'll be the day, right? That's the dream. And then also just to kind of piggyback on that, while I'm on that train of thought, if we do come up with a nice solution for these situations, for these contaminants, for these disasters, I really don't want to see a situation in which companies or just entire industries are now have an excuse to do, to continue business as usual, right? It's like, well, who cares if we spill oil in the Amazon? Who cares if we do all this stuff? Because we got fungi to back us up. That's definitely not the approach. I want my job to disappear. I don't want anything to do with this stuff, right? In the meantime, we do have this opportunity to align ourselves with fungi because they're, incredible species or incredible organisms. They form an entire kingdom all on their own. If y'all are completely new to mushrooms, just know that they're not animals, they're not plants, they're not bacteria, they're their own thing, right? And they're able to do so many unique things. But just to go straight into it, why work with fungi? Why do we even consider fungi for this stuff, right? It seems so crazy that we're going to be playing with mushrooms, right? As I was once told by some professors after I graduated and I came back and I pitched them the idea. They're like, why do you want to throw mushrooms at these oil spills? It's not going to do anything. All right, we're not doing that. We're working with fungi. We're working with the vegetative state of fungi. We're working with mycelium. And as such, fungi are really good at degrading things, specifically degrading organic constituents in nature. They are nature's recyclers. They've been doing this for millions of years, right? So we can trust that they can do a good job at that. In order for them to do this, they have this really unique system of producing these exudates, metabolites, enzymes, and I say these different things because they kind of all are interchangeable, but they also all come out at the same time. Fungi can release these things into the environment extracellularly, right? So they can actually produce these compounds. Well, from now on, I'll call it my favorite phrase or my favorite way to refer to these things as a biochemical cocktail, right? It's full of so many things, right? So they're able to produce these biochemical cocktails, release them into the environment, and degrade their food externally, and then absorb the things that they need to grow and continue their life cycle, right? Nowadays, everybody's getting into mushrooms. It's becoming really popular, and that's good because these mushrooms that we want to work with, these fungi that we want to work with to do these really crazy things like micromediation, they're relatively easy to cultivate, right? Nowadays, you can do so much with liquid culture, with producing all kinds of spawn, grain spawn, sawdust spawn, you name it, right? We can grow it in buckets, we can grow it in plastic bags. Hopefully not for very long because we can't really do a whole lot with plastic bags. But we can grow these mushrooms. We can expand the mycelium of these fungi in so many different ways that it's become relatively easy, relatively cheap, and very accessible. There's so many resources online. You know, organizations like CTMS are paving the way for people to come in, welcome them into the world of mycology, and teach them the basic skills to do this stuff, right? And up to this date, it may change with further research, but up until now, what we are seeing is that in the process of breaking down these contaminants, we're really getting some pretty harmless byproducts, right? In the form of water, CO2, and maybe some reduced versions of these organic pollutants that can maybe then secondary decomposers, and then a whole sequence of other organisms can come in and then really get the stuff broken down entirely, right? So keep that in mind, is that right now we can say that they produce harmless byproducts, but that's considering that they're going to be pairing up and partnering up with other organisms that are going to come in and then altogether get rid of the problem. And then obviously we've got a lot of scientific support. This is a field, micromediation is a field that has been looked at ever since, at least a few decades, if not more than that. And there's a lot of publications out there. Unfortunately, a lot of them are at the lab scale or small scale, or if they do make it to the field scale, which I'm going to show you some examples, they're not necessarily replicated or followed up with, or very easy to understand if you read the publication. So these are the reasons why we work with fungi. But let's just get straight into it. What does micromediation look like? I'll give you some examples. We've got our typical oil tanker, oil truck, transporting oil. It can be diesel, it can be different types of gasoline, it can be crude oil, whatever the case may be, right? These are traveling across our nation. If they're not on tankers or trucks like this, they may be on pipelines, right? Whatever the case may be, we typically see accidents happen, right? A pipeline may burst because of all the pressure that is needed to transport oil from one end of the country to another. In between, there are pumping stations, which kind of give it a boost so they can make it to those destinations. And these trucks, they also travel a long distance, so they can suffer. They can be they can easily flip over, like we're seeing here, right? So let's say we have this truck, it's transporting thousands of gallons of diesel, and it gets into a car crash because the driver is really tired and runs a red light, and so on and so forth. What do we have, right? We have a spill. In this spill, we have hydrocarbons, maybe we have some hazardous chemicals, but now we've got a situation that we have to come in and intervene, right? If we don't do this, it's going to affect ecosystems, it's going to affect other organisms, and then it's just going to be a chain of consequences down the line, right? So we've got contaminated soil. Let's just make it easy like that. We want to intervene by, applying this micromediation strategy, right? So how do we do this? We don't throw mushrooms at it. We don't go to the store and get portobello mushrooms or cremini mushrooms, or go to the farmer's market and buy some really juicy, delicious lion's mane from Hi-Fi and throw that on the oil spill so they can absorb it. That's not how it works. We work with the vegetative body, the vegetative state of the fungus, which is the mycelium. And in order for us to inoculate or treat these contaminants with mycelium, we need a substrate, right? My good friend Peter McCoy calls the substrate a battery, right? It's what's going to feed on as it's doing its work, which is to secrete these powerful enzymes within this biochemical cocktail. And then down the line, it's going to attract secondary decomposers and other microorganisms that are going to come in and assist in that job, or take it on and continue the chain of degradation. So how do we do this? Like when we talk about the mycelium phase of things, right? How do we obtain mycelium? How do we expand mycelium? This is the part that we're going to talk a little bit about cultivation, but then again, we're not going to go into too much detail. But this is a very typical scenario, right? We want to collect fungi from the local environment. That would be ideal. If you happen to have fungal strains from other contaminated sites, that would be preferred because they're already trained or they're already exposed to contaminants. But let's just keep it simple. We collect fungi from the local environment because they're going to be adapted to that place, right? We can isolate them under sterile conditions, right? We can take them to the lab and obtain a pure culture, right? So now we put it into cultivation, right? From there, we can expand it, right? It's the name of the game here. It's mycelium expansion, mycelium running. We are transferring the mycelium from one substrate to another. And like I said before, this has become relatively easy nowadays. We transfer it over to substrates, increasing the volume of our material, our biomass. We can even take it up to a scale that maybe it's not practical for the home cultivator, but we can use these large containers and grow it on things like wood chips, straw, cardboard, coffee grounds, right? We can do a mix of all these things that are easily accessible and are typically byproducts of some other industry, right? We got to think large here. And like I said, these techniques are pretty easy nowadays. If you can get your hands on some sterile cultures on petri dishes, get some grains, get some rye berries, get some, anything that you want to grow it on, whatever particular substrate you prefer. Get yourself a little pressure cooker, get yourself some jars, and you can start working with that, right? Nowadays, you can work with liquid culture. That makes things a lot easier. But just kind of keeping with the typical flow of things, we go from a petri plate or a wild mushroom, we transfer it over to a petri plate. From there, we expand it to grain. From grain, we expand it to more grain, and from more grain, we expand it to bulk substrate. Right? That's our typical flow of mycelium expansion. And like I said, we can grow it in pretty large containers. This is super cool. We used to do this at the research station all the time. We also happen to get really large fruit bodies, which were awesome to have. Okay, so now that we have contaminated soil, we collected, we put it into a pile, right? We have our mycelium expanded on substrate, and we've got tons of it, right? Now we enter the workspace. Notice this guy is dressed in full PPE gear, right? Respirator, helmet, visual vest so that people can see you. I mean, the whole thing. Pretty much what I was wearing at my spill sites when I was a hydrogeologist, right? And this is important to keep in mind, right? Like when we talk about micromediation, we're talking about dealing with some pretty toxic stuff. So we want to take care of ourselves, right? As we take care of our planet, we want to take care of ourselves, right? So in these 2,000 pound bags, what we're seeing is a mix of our mycelium, right? Our fungus, along with its battery, the substrate. So you can see here that we've got a mixture of spawn with another substrate. Again, this is large scale stuff, this is industrial scale stuff, but we can dump it onto the pile, mix it, which is not very different from what we do in the lab, right? We take the jar of grain spawn, we take our bag of pasteurized or sterilized substrate, we mix it, and we want the mycelium to cover as much of the volume of that substrate so that we give it the best chance of expanding and eventually colonizing. Once we have it all mixed in, we want to give it the proper conditions, the proper care. We want to love our fungi. We want to love our mycelium. We want to give it the best chance to grow, right? So we cover it. We make sure it doesn't dry out. We put in some irrigation lines. We aerate it. Maybe we mix it every once in a while so that the mycelium is happy and it's doing its job, which is something that it does naturally, right? In this case, we had an oil spill. And with the simple application of Mycelium, in 25 days, we had a reduction of about 47% of total petroleum hydrocarbons, 35% of polyaromatic hydrocarbons, which I'll talk to you about in a little bit, but just think of them as more toxic. And then we had a reduction of cadmium and lead, which are heavy metals that can cause a lot of problems, not just to us, but to the environment at large. Keep these numbers in mind because this is important, right? 47% is not 100%. 35% is not 100%. So what do we do with the rest of it? Is this successful? Are we done here? Did the fungi do a good job? You know, these are really important things to be asking at this point. And is this the best way to go about it, right? What are we trying to achieve here? These are the questions that I want you all to start thinking about. Let me give you all another example. This is over at Fungi Farm in California. My really good friend Levon was working with an old I think it was a parking lot. They had old trucks and they were spilling diesel and gas onto the soil. So once again, we scoop it up, right? We scoop the soil up, we put it into piles. In this particular case, I was kind of blown away by their approach. Instead of mixing the mycelium with a substrate and then introducing it in those big bags, like in the last example, what they did was take the same approach, right? Mycelium on a substrate or a battery. But in this case, they decided to go with 1,000-foot-long rolls of burlap that they pasteurized and they inoculated and they simply rolled them out, right? So this is getting pretty innovative, right? So we can create layers of this myceliated burlap sack or myceliated roll and then create layers. We don't have to work so hard in mixing it. But you can see that there's some challenges and some limitations to this as well, right? But just kind of giving you another example of what's been done at a relatively large scale. In a not so large scale, hold on a second. Fungi Farm on Instagram. Yes, Ali, thank you. Sorry, I just noticed there was a message, so I got excited. Cool. So this is a thing that we did at the research station. We collected dirty rags from the Austin Yellow Bike Project. As you can see, they are dirty, right? They've got grease, they've got oil, they've got all kinds of things on them. We didn't pasteurize them, we didn't sterilize them. All we did was layer them with sawdust spawn. In this case, we used spent sawdust or spent fruiting blocks from hi-fi, meaning that they've already fruited once, but the mycelium is still very much alive and happy and ready to do more work. So as you can see, we simply layer them, which is no different from what Levon did at Fungi Farm and what the guys at Polypop in France, they mix it all in, but it's a very similar thing, right? We want to get the mycelium in contact with the contaminated stuff so that it can get to work, right? After a while, we saw that the mycelium was starting to colonize the dirty rags. We even had fruit bodies come out of this project. Now, here's the other thing I want you to think about. Are these mushrooms fruiting from the sawdust alone? Are they fruiting from the rags? Are these mushrooms now full of contaminants? Have we done a really good job at breaking this stuff down? Are we successful? You know, are these fungi successful at doing this stuff? Right? These are the things that, once again, I want you to all think about. Nonetheless, it's really cool to see mushrooms fruit from this kind of experiment because of a couple of things, right? It could mean that it's a successful project because the mycelium took off on this material and it's happy and it's producing fruit bodies because it wants to reproduce and it wants to go and spread its life somewhere else, right? Maybe, maybe not. What's interesting is that from this fruit body, we can now obtain strains, right? We can collect spores and we can isolate individual strains that are now adapted or that have been exposed to this environment, to this contamination, maybe just the fumes, maybe just the things that they're breathing, or very likely they have adapted to breaking down, or I should say they have adapted themselves to build up tolerance and maybe how can I say this? Like maybe they're better at degrading this stuff than they were before, right? So we can do a lot with these fruit bodies, even if the rags are still there, even if they haven't gone away. But trust me, they did go away. I'm just showing you this picture as an example. So yeah, these are the basic scenarios. This is what we've done so far. You may think, well, you know, at the end of the day, don't we want fungi to do the work on site? Do we really have to scoop up the soil? You know, doesn't it seem so, you know, machine heavy, equipment heavy? You know, is this going to be the future of micromediation? You know, we'll cover some of that stuff later. So at the same time, you know, if we know that fungi can do this so well, right? We know that fungi can adapt, they can degrade externally. We know that there are harmless byproducts. We know that, you know, et cetera, et cetera. We know all these things. What's keeping us from doing this? What's kept us from doing this? What are the main obstacles, right? Well, like I said before, one of the main obstacles is that there is no follow-up to these large-scale studies, right? Where are the replications or the replicate studies at least? Where are the follow-ups, right? Where are the publications that tell us all the things that happened? These are common questions that I've been asking myself for the past few years, but it comes down to that. There's a lack of follow-up and replication of these experiments, right? So in this case, you know, going back to these ones, you know, I mentioned the concentrations. Did we try doing something? You know, did we try applying a secondary decomposer? Did we try bringing in some, you know, throw in some compost seed to bring in some beneficial bacteria that can do to do the rest of the job? No. In this case, did we, you know, and I can go on, and I really don't want to shit on these projects because they're really cool. I forgot to mention this one with, you know, the Paul Stamets talk, you know, the Paul Stamets TED Talk, this project that, you know, launched my interest in micromediation. You know, there's this, you know, once again, we got these big piles of contaminated soil. We mix it in with sawdust spawn, and then we see the mushrooms fruiting. And the mushrooms obviously attract bugs. The bugs attract birds. The birds bring in seeds. The seeds germinate, and we've got plants growing, and now we've got this nice living ecosystem, a thriving ecosystem on this pile. Is this successful? You know, are we done? Is our job here? Is our job done here? I encourage every single one of you to look up the actual publication of this study, in which the actual analysis will say that, you know, it's kind of inconclusive. We don't, you know, there's not really much of a difference in the actual concentration of these contaminants in the before treatment and the after treatment. So did we do a good job? Are we done? Because mushrooms fruited? You know, these are some of the questions that we need to be asking ourselves. Second thing, you know, a lot of the research that has been conducted, like I said before, for decades now, has emphasized on pollutant degradation. You can find studies on dozens of strains or dozens of species of fungi, not just the wood decomposers, but other fungi as well. And you can see that they've been tested against dozens of pollutants, right? Anything from hydrocarbons to agrochemicals to pharmaceuticals to dyes and other things, right? We're not going to go into all the different categories of contaminants, but we have focused so much on whether fungi can do this in the lab without giving equal emphasis on large scale production of mycelium, which eventually we're going to need to put out into these sites, right? Because cultivation, you know, mycelium expansion is expensive. I mean, if you're thinking about it at this scale, you know, imagine, for those of you that live in Austin, for those of you that know hi-fi, just imagine the entire hi-fi mycology operation dedicated to just producing spawn for micromediation treatments. You know, I mean, that's the scale that we need to be at in order to do this kind of work with these examples, right? And so there hasn't really been that much emphasis on it. This is my chance to talk up a little bit of the research station, because that was the focus, right? The focus there was to bring in the spent blocks from hi-fi, and nowadays we're working with small hold to get these to get mycelium expanded, right? To get bulk mycelium in the hands of whoever wants to come and get it and run these experiments, or for ourselves to do it, right? One of the cool things that we did at the research station, or I should say at Circle Acres, right? Not just the research station, but the whole site at large, was receive these agricultural byproducts that would eventually be the food source for our mycelium, right? Truckloads, 40-foot trucks would dump, wood chips and other things that we can convert into substrate for mycelium expansion, right? Once again, I just want to highlight the work that volunteers have done for the past 6 years now, doing this work, wheelbarrowing the substrate over to the research station, pasteurizing, mixing, inoculating, putting it into containers, taking it to places like the Whole Life Learning Center, giving it to people, putting it in the hands of people so that they experience bulk mycelium, and they're not stuck with one jar, one petri dish, one bag of sterile stuff that is going to fruit once and then you don't know what to do with it, right? The idea here is that we want to inoculate people's minds, get the mycelium into people's hands so that we can get used to working with fungi, get used to working with mycelium. This is a big task that I set out to achieve at the beginning of the research station, and by now, it really warms my heart to see people like Sam, starting a business from this kind of work. It's amazing. I want to see more of that. Composting it, right? We have compost piles at the site too, and this just increases the biomass, not just of the mycelium that we're going to be using for the applications, but to increase other secondary decomposers and other beneficial microorganisms that are going to pave the way for the later stages of the degradation. And then the last thing, the third thing of these obstacles that I want to highlight is the quantity of mycologists and the type of mycologists, right? I mean, it's so cool to see everybody jumping on the hype, on the bandwagon. I encourage it, I welcome it, great. But what are we doing? Are we all just focused on growing mushrooms? Are we trying to take this to the next level and coming up with, programs, and maybe in the academic setting, come up with degrees for applied mycologists, right? Micromediators, right? Can we create a situation in which there are mycologists that are dedicated to mushroom cultivation, to taxonomy, but also to micromediation, and feel proud of that, right? So one of the things that I focused on as well during my time at the research station was to inoculate the next generation of mycologists, right? We welcome schools, school programs, after school programs, I should say, homeschool kids that, the parents reached out and they wanted to give their kids a fun activity to do. And, it doesn't look very different to what some of y'all did, some of y'all grown-ups, adults did during my workshops. It's get your hands dirty, mix in the substrate, break up the spawn blocks, and then bag it up, right? Take it home, observe it, watch it get moldy, because that's typically what happened, or watch it grow some really awesome mushrooms and eat them, right? And yeah, so through that, developed a whole program of workshops and classes, taught so many people. Yeah, I can go on and on about that, but y'all have probably heard me talk so much about this stuff and have actually seen it that I'll just kind of go right ahead and keep going, right? So happy to see all the smiling faces take home blocks of mycelium. That was always such an entertaining thing for me. Cool. And then the last thing is lack of project sites available to citizen scientists, right? Playgrounds. Where can we experiment? Where do we have labs to come in and practice, and how do we get good at this stuff? Because if we look at large scale, industrial scale spill sites, there's no way that a regular person is getting in there. There's so much legislation or regulation involved with it. You need to conduct studies, you need to wear the right gear, you need to have the right certifications, you need to have a 40-hour HAZWOPER training from OSHA. Like there's so much that you need to do, but at the end of the day, just there's no way that you'll get involved in these projects unless you have all these requirements. So how do we get good at this stuff, right? How do we practice, right? Well, through the, with the examples that I gave you all, the small scale stuff, the dirty rags, the spilled oil in your driveway. I mean, those are really good places to start, but there's still a lack of places to do this stuff, and start getting good at it at the larger scales. So once again, it's my chance. It's the moment to kind of talk up Circle Acres and encourage everybody to come out to this site that has a very long history of abuse, of mishandling, but ultimately of recovery, right? The work that has been done by the Rhizome Collective, by Ecology Action, and ultimately by the MICO folks, either through MICO Alliance, through the Central Texas Mycological Society, or nowadays the Mycological Research Station, has been fundamental to getting a site dedicated to this kind of work. And so if you're in the Austin area or in the Central Texas area and you want to come up with a project, if you want to see others do or help others and assist in these projects, come out, right? There's so many things to this site, right? It's got a wetland, it's got a former landfill that's been capped, it's got trails, it's got a number of creeks entering it, it's got runoff from the subdivisions, it's got a golf course nearby, it's got the university nearby. So there's so much going on with this site that I encourage all of you to come out, reach out to Eric, reach out to Ecology Action, and get to work, right? Let's get good at this stuff. This is the wetland. So yeah, let's see. I see another message. Cool. Ali's just sharing information. I get excited about these things. Cool. So what are we looking at here? What's going on here, right? We've got a typical log that's fallen, right? Either dead or in the process of dying. And we see these mushrooms come out, right? These are oyster mushrooms. So right away, I want to jump straight into it. We are working with saprophytic fungi, saprobes, decomposing fungi, and more specifically, wood decomposers or wood rotters, right? In the previous image, in this image, we were looking at oyster mushrooms, which are edible, delicious. I encourage everybody to find yourself some mushrooms in the wild or cloning them and working with oysters because they are the easiest and most accessible. This is a really nice picture that I took down here in the peninsula of a Schizophyllum commune, the split-gill mushroom, which again is a wood decomposer. Lentinus crinitis, Turkeytail, right? We got so many varieties of wood decomposers. It's I wouldn't say infinite, but we do have thousands of species that we can work with, right? And they all kind of do different things. They focus on different types of wood. They focus on different types of trees, and they look different. They can either be easy to clone. The chunky oysters. They have a lot of tissue that we can use to clone. The turkeytails not so much. We have to go at the base. At that interface between mushroom and bark, there's a really nice area there to clone the turkeytail, or cutting it in half and getting real close and personal with it. But whether it's oysters or turkeytail or lentinus or whatever it is, we're pretty much seeing the same thing happening, right? Obviously, we're not looking at a tree here. We're just looking at a very general schematic. But what we see here is a mushroom, right? The fruit body, what we see and what we interact with the most. And underneath is what we call the vegetative part of the fungus. the hyphae, or I should say the mycelium, which is composed of hyphal strands or hyphae. And what the hyphae does is they essentially break down organic matter, converting organic matter into nutrients, and those nutrients, become the source of food, the source of energy, so that they can continue to grow underground, inside the trees, inside the wood, so that eventually, once it's ready to go somewhere else, inoculate other substrates, it'll produce a really nice mushroom, very attractive, edible, maybe not edible to us, but edible to other things. And that way the mushroom can produce gills or pores or sacs or something else to release spores. And that's essentially the life cycle of our basidiomycetes, or our wood decomposers. And in order for them to break down the organic matter, once again, they release these enzymes or this chemical cocktail, so they can do it externally, right? They are sweating this stuff out, breaking down the organic matter externally, and then absorbing whatever it is that they need. When we look at wood, we're looking at three main components. Obviously, there are other things, but we're really looking at three main things. When it comes to micromediation, we have to focus on one of those things, which is lignin. Lignin is not actually something that's fixed. It's this kind of random polymer that exists all throughout nature. It can vary from tree species to tree species, but also within different parts of the tree, lignin can look different. So it becomes this really abstract thing that exists in nature, but it serves a very specific function, which is to protect the cellulose and the hemicellulose, which we can just consider carbohydrates for other organisms that are going to break it down, right? So the lignin acts as this interface, as this protective layer that without white rot fungi or wood decomposing fungi, they'll never be able to tap into the cellulose and the hemicellulose, right? This is a very general statement, an oversimplification, but if anybody wants to read up on the development of the evolution of wood decomposers and how they came about, how they developed the ability to break down lignin, I've got some papers, and some of y'all have heard me say this before, but on the geologic time scale, the timing of fungi developing the ability to break down lignin matches the time in which we stop producing fossil fuels, right? It's a reason why they are non-renewable. It's because fungi eventually were able to tap through the lignin, break it down so that the wood material, the biomass, wouldn't just get buried, compacted, and then ultimately get turned into things like natural gas, right? So if any of you all are interested in that kind of stuff, reach out to me. I'm happy to share some papers. But everything that we talk about when it comes to micromediation comes down to this, right? Especially when we're talking about wood decomposers. Notice that I'm not bringing up mycorrhizal species or mycorrhizal fungi, endophytes, parasitic fungi, which can also play a role in degradation. Here we're going after the really intense stuff, the really recalcitrant contaminants. And in order for them to do this, they have to break through lignin. Once again, I'm not going to go into all the chemical stuff, all the chemical background. Just visually, look at lignin. Look at the composition of lignin. It's made up of all these different aromatic rings. The rings are really hard to break down. It's only fungi that are able to do this. And then when we compare the structure, the chemical structure of lignin, right, these large aromatic rings, they look almost identical to our aromatic hydrocarbons, our pollutants that we deal with when we have an oil spill from an oil tank or from a pipeline, right? The polyaromatic hydrocarbons. And in particular, we're talking about the 5-ring, 6-ring PAHs, which are so hard to break down in the natural in nature. And so you can kind of start to see where we're going with this, right? How it is that we came about developing the field of micromediation is we observe nature. What do fungi do? Why do they do it so well? and how do we translate that into some of these common problems in our world, right? And it's by observation, right? So let's get into a little bit of this, these wood degradation processes. We have different types of decay. We have different types of degradation of wood, and they're typically categorized by how they degrade wood, right? How do they leave it behind after they're done degrading it? Their general biology or ecology as well, and whether they're primary or secondary colonizers or secondary decomposers. We have soft rot at the very bottom of the scale, maybe the least important, but still very relevant and very widespread. Is we have soft rot, right? They degrade cellulose and hemicellulose. These can come down, down the chain of degradation. They typically look for high water content, high nitrogen content. This is what we see found in window frames, wet floorboards, fence posts, you name it, right? Anything that we have a lot of contact with. They become very wet, it becomes very wet, very spongy. Just one second. Here we go. Very wet, very spongy, and they can, there we go. And they don't specialize too much, right? So they can break down a lot of different things. Cool, and then we step over to the brown rots, which we've seen before as well. These can also only degrade cellulose and other carbohydrates like hemicellulose, and they leave the lignin intact, right? Of all fungi or of all wood decaying fungi, we only have about 6% of them being brown rots, and they are fragile, powdery. They have these brown cracks on them, and you'll be able to identify them not just by the brown color, but because they form these cubics or these little cubes once they're done degrading the wood. And of course, we have the heavy hitters, the main fungi that I research and that I focus on, which is the white rot fungi. These can degrade all components of wood. They can break down lignin, cellulose, hemicellulose, and almost everything in between. What happens is that it's an oxidation process, right? It's an aerobic oxidation process that causes the wood to essentially take on this white or bleached appearance, which is why we see it often bleaching the wood. It forms these long strands. And it includes all of our saprophytic fungi that I mentioned before, anything from oyster to turkeytail to other pathogens as well, like the honey mushroom. They can do, they can break down wood in this way as well. And lignin degradation nowadays is I don't even want to estimate it, but it's at least $1 billion industry. People are looking at lignin degradation and conversion for their use in things like chemicals, right? Fuel, electricity, right? So we're really looking at an industrial scale process that involves pyrolysis, thermolysis, oxidation, combustion, so on and so forth. We're not going to go into all these details, but just know that there is a whole industry behind wanting to break lignin down and turning it into something else, right? So once again, we come back to this idea, right, of these fungi being easy to cultivate, right? And the reason why they're easy to cultivate, or relatively easy to cultivate, is because the mushroom itself is made out of hyphae. It's made from mycelium. It is mycelium, right? It's just taking on this other expression. It's a superficial expression of the mycelium. It's what I like to say about it, right? And so we see in this image here, the mushroom body itself is composed of hyphae, and because of that, we can take it into the lab. We can extract some tissue from it. We can put it onto a petri dish. We can clone it, right? This is a process called cloning cloning the fungal strain or the mushroom strain. And this is something that I do all the time. This would be a nice little opportunity to show you some of the things that I work with. This is a nice little polypore. This is a wood rotter, white rot. This is a red belted polypore. Hope you can see how big it is. It's about the size of my face. I've got some Ganoderma applanatum. Again, massive fungi. Some more red belted polypores, and so on and so forth, right? These mushrooms are hard as wood, but like I said, like I brought up with the turkeytails, there's a really nice area in this interface between the mushroom and the bark and the tree that we can take a piece of tissue out and clone it, right? If you want to take it from any other part of the fruit body, feel free to do that. If you can cut through this stuff, like I said, it's pretty hard. You may want to do it beforehand instead of trying to do the actual cutting and the actual stripping of the piece in the lab, because it's really tough, right? Once we do this, though, we have an exact clone of this, right? We are not collecting spores, so that we can isolate different strains. We are cloning the exact strain that we found, right? So I'm often out in the wild looking for these kinds of mushrooms because they are not just really badass at degradation, but they also happen to be highly medicinal, right? We can turn them into tinctures, right? These are some of my friends down here in Mexico making different kinds of tinctures with these polypores. We've got a blend of 12 different polypores that we can throw in together, and all together can form this really awesome, really potent medicinal formula for all kinds of things, which I'm sure someone else will cover in a different talk. Again, going back to this idea, easy to clone or easy to cultivate, well, we know that we can use things like cardboard, right? Some of you all that have come out to my workshops, you have cloned these mushroom species or expanded this mycelium over to cardboard, rolled them up. We call those Mycelium burritos. They're delicious. You all should have some. Put some salsa on it. No, I'm just kidding. Don't do that. We can grow them on all kinds of household waste materials, filters, toilet paper rolls, spent coffee grounds, you name it, right? You can do so much with this stuff. And once again, I'll leave it up to the Central Texas Mycological folks, and everybody in Austin and everybody else, wherever you are, to cover this stuff because it's so accessible, and I encourage everybody to do this. And I won't really go to too much detail about that. Nonetheless, once you clone it, you can pretty much expand it infinitely if you are, if you got some good techniques and you can replicate a process. It's all about repetition. It's all about getting really good at it. You can pretty much expand it until the end of the day, until the end of your days, right? Infinite. There's some strategies there. You got to change the nutrition. You got to change the different parameters. You can pretty much keep that strain going for a really long time. I've got some strains that I've kept with me for 4 years, 5 years now, and I'm so happy that I got good at this stuff because now those are my go-to. Yes, I love cloning. Yes, I love going to other places, like contaminated places, and finding new strains. But I also like to rely on they're not even mine, just the ones that I've been playing with for a long time. And like I said before, it's gotten to the point where we can work with liquid culture. Liquid culture is a really awesome technique to expand mycelium without the need of a very sterile environment. We can do this relatively cheap with water and sugar, some minerals, a syringe that you can buy from the pharmacy, and you're pretty much good to go, right? You can sterilize the nutrient media, inoculate it with spores, you can inoculate it with more mycelium, or buy a syringe online. $10 will get you a nice starter culture that, once again, you can just keep going. You can just expand it infinitely, right? So kind of going back to the micromediation stuff, this is something that I've been doing for a while. And I started doing this back in 2014 when I was down in the Ecuadorian Amazon basin, looking at a really large scale oil spill. And we found these logs, right, covered in, like, soaked in oil. And these mushrooms were fruiting from these logs that were soaked in oil. And you start to wonder, like, what the hell is going on? Like, why would this mushroom do this? Or why aren't we working with that mushroom? That's a better question. Like, why don't we go find these mushrooms? Why don't we get these mushrooms that are already tolerating, at the very least, tolerating the overall environment, right? As you walk up to these spill sites, you start getting a headache, you start breathing in so many things. Once again, you got to have the right protection. Otherwise, you start hurting yourself, right? But these fungi are there, they're thriving. Look at these turkeytails. Look at these, like, polypores that are growing on this log. They look no different from what you would find at a mushroom farm growing from an inoculated log. They're thriving, they're happy. So what's going on with them? Once again, we go back to this diagram. The mushroom is just a superficial expression. Everything is happening underneath, everything is happening out of sight. But we've gotten a really good sense by now of what happens in these underground spaces, right? The mycelium, that's composed of hyphae, which are individual strands that all together form mycelium. It's not one organism that is just growing, that if you cut it, it dies. You can actually expand it infinitely. The hyphae is releasing enzymes as part of this chemical cocktail that breaks down organic matter in order to get nutrients so that it can continue its growth. So we get into enzymes a little bit, right? These enzymes are catalysts. I'm sure you all remember, chemistry 101. Enzymes, what they do is they speed up a chemical reaction, or they essentially speed up a chemical reaction, or they make it happen, right? When we talk about lignin degradation, which can be translated into micromediation of toxic pollutants, we're really looking at 3 different enzymes. These are altogether known as lignin modifying enzymes or lignin degrading enzymes, right? Lactase, lignin peroxidase, and manganese dependent or manganese peroxidase, right? Notice that all of these need something, right? They need something to react on, right? In this case, they use hydrogen peroxide as a co-substrate, right? Meaning that they're going to break down the hydrogen peroxide, right? Peroxide, peroxidase. Anytime you see the ASE, it's an enzyme that is going to do the work of whatever it is, whatever the other name is, right? Peroxidases work on peroxide. And these enzymes, they're not subtle. Like, you see it everywhere. You see mushrooms, you see fungi or mycelium producing these enzymes all the time, right? I give this example quite often of this contaminated bucket, right? We have trichoderma, a really nasty yeast that or mold that takes over the substrate, right? That we don't want it to do that. Even though trichoderma is really good for soil and really good for other applications, we don't want it in our buckets because we don't want our fungi to waste energy fighting it off, because that's what it does. If it's strong enough of a strain, it'll fight it off. And the way it's going to fight it is by producing a novel cocktail that's essentially going to attack that contaminants, right? Not all fungi produce specific cocktails. They can produce non-specific enzymes that can attack a lot of different things, and not all fungi will release all 3 different enzymes. It depends on nutrition. It depends on their actual, evolutionary traits, and different parameters like pH, temperature, and then substrate, which is what they're growing on. But so in this example, I isolated that bucket from the rest of the buckets in the greenhouse because it's contaminated, and I don't want the mold to expand because it does that very well. So I isolated it, left it alone for a couple of days. And when I came back to it, I noticed that there were these droplets on the surface, and I saw the mycelium was now growing on the trichoderma. It was fighting back. It was defending itself and ultimately breaking it down through this extracellular enzymatic production system. Well, what does that tell us about that chemical cocktail or the biochemical cocktail, right? It's got to have something in it that's killing off the trichoderma. So if we know that trichoderma is prevalent and it's commonly found in our environment, can we extract this cocktail that we know it works on trichoderma in this bucket and use it in other applications, right? Can we prevent trichoderma from appearing on our buckets if we spray it, if we rinse it with this cocktail? Does that do anything, right? These are really interesting questions. Before I get too ahead of myself with enzyme stuff, what we can do with this stuff, what we can do with not just the breakdown of the contaminants, the actual understanding of what enzymes are present and what they're able to do, we have to start getting really good at observing and measuring mycelium and its behavior on different nutrient media and on different containers, different substrates. But yeah, I mean, I'm kind of repeating myself, but we essentially have to observe and measure and quantify mycelial growth, mycelial behavior, right? And so these are things that are called screening tests, right? We set up a kind of like a pre-filter, right? If we think about micromediation strategies, this is the very first phase. We have thousands of species of fungi in nature. We're not going to be able to test all of them against the dozens and maybe hundreds of contaminants that we're going after all at once. That's too expensive. That's too time consuming. So how can we filter out these species? How can we decide and separate the ones that we want for a particular application from the ones that simply don't do a good job at being the first responders, for example? Maybe they come in later. Maybe they come in as secondary decomposers, right? There's a lot of different ways to approach this, but screening tests are essential if we're going to speed up the process at which we are selecting candidate species for micromediation applications. So I want to present to y'all a very quick, easy test to compare different strains and different species, different strains of different species, or between different species. This is growth measurements, right? For a long time, I've been thinking about this issue of measuring growth. You know, how do we actually put some numbers to mycelial growth on petri dishes, on grain in a jar, on bags of sawdust, and ultimately on an actual contaminant or an oil spill, right? How do we measure its growth, which will ultimately tell us how successful we are, right? Well, let's just get straight into it. With this particular strategy, what we are doing is we are placing our transfer wedge, right, from another petri plate, from an agar plate. We're transferring it over to our screening plate and placing it right in the middle, right? Then what I do is I put a grid that's transparent and it's composed of different squares that I know what their size is. In this case, they are 1 millimeter by, or 1 centimeter by 1 centimeter, one centimeter squared cube, right? We have an area of 1 centimeter squared, right? What I do is that I put my transfer in the middle, and then over time, I'm counting the number of squares that the mycelium is colonizing, 50% or more, so that I can say that it's colonized, right? What is this going to do over time, right? We go from day 1 to day 3. We see that the mycelium is expanding. It's colonizing more and more of its substrate, but more importantly, it's covering more of these squares, right? So over over time, we know the size of the square. We know how many squares make up an entire petri dish. So we can start getting some actual numbers. We can actually start quantifying growth rate, right? In this particular case, on day one, which is the moment at which the mycelium jumped from the wedge, the transfer wedge, over to our fresh substrate, our fresh nutrient media. That's day one, which typically happens after 24 hours of transferring it. We can see that we have two of those squares, or one square, fully colonized, meaning 50% or more. That represents 3% of the entire plate that is now colonized. We jump to day two, we have 11% colonization. Day three, we have 27%. And by day four, we're seeing at least 50%. We're seeing more than 50% of the plate be colonized, right? We can transfer this colonization percent colonization over to an actual rate, meaning that we can transfer this over to an actual number that says this particular strain of Turkeytail growing on a very traditional formula, which is PDA or MEA. It's growing at a rate of 2 square centimeters per day, right? That's not necessarily the case with this example, but just want to give you the idea of where we are going with this, right? If we can start putting numbers to this stuff, then we can take any number of strains of that particular Turkeytail species, or compare it to however many thousand species we have, and actually make comparisons that make sense, right? Because what happens when, instead of using traditional nutrient media or traditional recipes like PDA or MEA, we throw in atrazine, we throw in hydrocarbons at different concentrations? How do we determine success? How do we determine degradation, right? When we do this stuff, we can actually compare in a significant way, and not just for ourselves, but for the community at large. Does it make sense for us to just say, well, we clean up an oil spill because we saw mushrooms fruiting from the pile of dirty soil? What does that tell us, right? It can mean a number of different things. When we start looking at numbers, when we start looking at the actual things that are happening, we can compare and contrast and make solid conclusions from this work, right? So this is just the pre-filter. This is just first thing we do. The first thing that I do when I collect a fungus, I like to get its growth rate and compare it on different nutrient media. Maybe I'll deplete it of a particular nutrients, stress it, and then go back to that original recipe and see how it responds, right? This is called response activity, or response time, response behavior, in response to a particular stress, right? Because, going back to what I said at the beginning, like, we can work with sterile strains, sterile mycelium growing in sterile environments, but when we take it out into the real world, it's not going to do a whole lot, right? It's going to be outcompeted, it's going to be overstressed, and maybe it won't end up being a successful installation, right? So these are the things that we can think big, but then act small, right? It's in these little details that we become good, and we become good at doing this stuff, but also good at communicating success and ultimately working with fungi in the right way. So getting into enzyme assays, right? We can kind of step up from growth rate on traditional nutrient media, PDA, MEA, whatever you want to use as a recipe. We can instead of using that traditional recipe, we can expose it to a contaminant at different concentrations and measure the growth rate there as well, so that we have comparative numbers now, right? So now we can say, Turkeytail, this particular strain of Turkeytail has a growth rate of 3 square centimeters per day growing on PDA, but on 1% concentration of spent motor oil, it grows at a rate of 1.5 square centimeters per day, right? So now we have something to compare, right? And this is just growth rates. And that in itself is already telling us a whole lot. not just within species, not just within strains, but comparing everything in between. Then we look at the actual mechanism for degradation, right? We talked about enzymes, right? We want to be able to observe enzymes, measure enzymes, communicate enzyme stuff, right? So one of the things that I've been working with a lot in the past few years, and I've shared that with many people, is this very simple protocol using Wycol, which is also derived from wood, and it's used in many different applications to give it, like, I think it's with whiskey or bourbon that they give it this kind of like smoky flavor or smoky aroma. I'm not entirely sure what they use it for. That's just what I read. But what I use it for is for these enzyme assays. And what you can see is a color change, right? Because what happens, going back to the enzyme slide, is that we have a co-substrate such as peroxide, hydrogen peroxide, which is H2O2. When the enzyme reacts with that, right, it acts as a catalyst for its breakdown into water and free oxygen radicals. Those free oxygen radicals then react with Wycol and produces a change in color, right? So you're looking at this on a petri dish because what I did was take my regular recipe, PDA, MEA, mix it with Wycol, and then just transfer the wedge, the transfer wedge over to it. And as it's growing, consuming the source of food, which, in our PDA, it's, potato dextrose, it's secreting or producing this biochemical cocktail that is composed of not just the enzymes, but the co-substrates like hydrogen peroxide, which we can just call metabolites or exudates, that all together react to do the degradation, such as degradation of lignin, right? Well, we can use these mechanisms, we can use these systems to our advantage, right? We're not really doing a whole lot. What we're doing is just, working with what fungi do naturally. And what they do is they produce enzymes, they produce co-substrates. Those co-substrates get broken down, and we're just simply playing with the product of that chemical reaction, right? O2 will react with Wycol, and we see this change in color on a petri plate. We can take a little ruler, or we can take the grid that I'm using and measure the size of that halo around our mycelium, and then start to compare it, right? And this is just a little spot check, right? Do we have lignin modifying enzymes such as peroxidases present? And then how present are they compared to other strains, other species, right? So once again, just a little spot check. Are these enzymes being produced? Are they being produced on this recipe more than this recipe? And then we can just take it to the next level of selecting our candidate species, right? We can do something very similar with Wycol and the enzymes. And instead of using petri plates, petri dishes, we can use something like a colorimeter, right? Because once again, what we're looking at is a color change, right? The intensity of that color change. So colorimeters and spectrophotometers, they measure that change in color because they send light through the medium, and then they measure the absorbance of that light through that media, right? So once again, all we're doing is creating tools, creating methods of comparison, right? Which will ultimately pave the way for us to scale up and reach our objective, which is to be successful at degradation. So I encourage everybody to look up open source tools such as IO Rodeo. They have this little Arduino powered colorimeter, or look at Public Lab out of New Orleans, and they have some other tools, open source. You can modify them, you can play with it and get some really good data, right? I won't bore you with all the data, but essentially you can measure absorbance of light over time. And again, in those little cuvettes, what I did was put in the raw enzyme exudate, the biochemical cocktail, mixed in with hydrogen peroxide and guaiacol, and then just track the color change over time, right? And what this gives me is a rate of change, right? A rate of change in absorbance, but then again, it's also a rate of that O2 production, right? So I forgot to include some of the other slides of the tools that I use, but essentially measuring O2, just straight up measuring O2 gas is another way that we can go about this. Measuring CO2, because as you know, as y'all know, basic mycology, fungi breathe O2 and they release CO2. So we can measure CO2 production over time on regular, on traditional nutrient media versus contaminated media. And that in itself will give us a rate of change because CO2 is going to be produced more depending on how or at the growth phase of the mycelium on that particular media. So it's another tool that we can track it. But once again, you know, without getting into all the details, it's the mindset. We are observing nature. We are paying attention to what fungi do. They breathe oxygen. They release CO2. They produce extracellular enzymes along with other exudates that I call biochemical cocktails externally that we can collect, right? And then we can do the degradation. And those are things that are just right in front of our faces, right? If we pay attention to nature, it's telling us everything that we need to do to develop the tools, to develop the methods, to develop the strategies to be successful at doing this kind of work. So again, biomimicry, paying attention, whatever you want to call it, fungi, nature is giving us all the answers. We just have to be willing to work with it and ultimately pay attention. If y'all have any more, like if you have any questions about this stuff, you know, we can go deeper into it. I'm happy to talk more about this stuff. Again, I didn't want to make it too boring with data. Like, this is not really what that's about. I really just want to get you guys and everybody here thinking about how I've been approaching this for many years and how I want y'all to approach this, you know? So just looking back at what's been done, kind of like on the mainstream side of things, right? 2005, we see Paul Stamets publish, you know, what is, you know, pretty famous book by now called Mycelium Running. In this book, you'll find everything from fungal ecology, you know, the different types of fungi, to mushroom cultivation, mushroom identification, extracts, tinctures. But in there, there's a really good section on micromediation that is easy to get through. It's not so technical. That actually was something that annoyed me at first. It's like, cool, these are really great stories of how wine caps or Stropharia rugosa annulata is able to break down or filter out E. coli. But how can I do it? You know, where's the manual? Where's the step-by-step instructions on how to do this stuff? You know, but nonetheless, it's a really good introduction, and it's good enough to get people inspired and good enough to get people started in the world of micromediation. A year later, we have the total opposite of this. We have Singh's Micromediation book, or Fungal Bioremediation. I've got it right here. It's a thick book. It's so technical, very technical. It references so many different publications. It's a really good reference to have for anybody that is out there looking to get started in this stuff. Micromediation. You know, I'm not going to say I read it every day, but you know, there are times in which I actually go back to it because it's just nice to kind of flip through a book rather than downloading PDFs online and staring at a screen. So yeah, that came out a year after, and then on the more accessible, so very you know, informative and starting to take it up. You know, starting to advance the conversation and the intricacies and the complexities of the stuff, we see Trad's book, right? Trad has been someone that I've collaborated for a while now. It's such a good book for, once again, everything from mushroom cultivation to identification, making extracts, growing morels, you know, taking mushrooms into the classroom. But ultimately, it's got a nice little chapter on micromediation. One of the things that excited me the most about this is that it actually breaks down the process of training and exposing different strains to different contaminants in a stepwise process, increasing the concentrations of the contaminant, and also working with enzymes. This is where I got most of my enzyme inspiration and foundation stuff from. So I can't really recommend it enough. This is a really great book. And then a couple of years after that, we see Peter come out with this massive thing, Radical Mycology, which once again kind of covers everything plus some, right? It's got spagirics, it's got psilocybin stuff, it's got mycorrhizal stuff, it's got some games in the back, it's got charts and forms that you can fill out or replicate and take out to the field. Man, I can't be thankful enough for Peter, to Peter for publishing this book. Obviously, it's still limited. It's not a manual for an engineer to take into the field and do micromediation, or for someone like me to go into the lab and really take it up a notch. But it covers so much, and it's got really good visual aids. It's got charts. It's got all kinds of things, right? Once again, I can't recommend it enough. This is a really good book to have. My friend Alex Dorr, published a little guide, a little handbook, not long after that, in which he kind of covers some of the things that I talk about with the enzyme stuff and some of these techniques and methods. But more than anything, this kind of compiled some publications that came out after these other books. And so it's really nice to look it up. I can't say it's a perfect book. Obviously, it's a first edition. Alex is no longer doing stuff with micromediation. He has gone on to run a really successful medicinal mushroom extract company. But it just kind of shows you the evolution of this stuff. And then that's it. Like, where are we at with this stuff, right? Who is publishing books about this? Where are we going with this, right? And so I put this slide because Juliana Furshi over at Fungi Foundation published it, or used the slide at the Telluride Mushroom Festival. I came into it and it just said, WTF, where's the fungi, or where's the fungus? And it's just like, what the fuck? What are we doing? What's next? What's coming next? And hopefully, I'm able to do some really kick-ass work here and maybe publish something in the coming years. I can't say that I'm much of a writer, except a technical writer, but I would love to see more of this stuff come out, to be compiled and pave the way for the next generation, because we've come a long way since Mycelium running, since Trad's book, since Radical Mycology, and definitely since Alex's book. But where are we at, And so with this, I just want to kind of wrap things up and say that anybody out there listening, all of you listening, that are interested in this stuff, right? Any of the things that I talked about today, reach out to me. I'm happy to talk more about it. I'm happy to put you in touch with the people that I mentioned, or the organizations that I mentioned, whether they be in Texas, in Mexico, South America, Europe. I'm not going to say I'm connected all over the world, but I've been reading enough publications to know where the hotspots are. And so if you're interested in this stuff, let me know. Let's have a conversation and let's publish some books. Cool. With that, I'll just wrap it up and open it up to some questions. And yeah, if y'all wanna reach me, obviously you can find me on Instagram. or just straight up email me at michaellyons@gmail.com, and I'm happy to have a conversation. So with that being said, I just want to say thank you for everything. And I'm happy to take some questions.
That was awesome, Daniel. Thanks.
And do you want me to leave the slides up in case anybody wants to refer back to the slides, or shut it down and have?
If you want to leave your contact info up there for a little bit, that'd probably be a good idea.
Cool.
And I'll just make us a little bit bigger on the screen, and that'll work just fine. So there, yeah, and if anybody has any questions, feel free to ask. One of the questions in the chat was with the, how do you cut those dried mushrooms? Do you cut them, or yeah, how do you do it?
Actually, something like this, I've used everything from a little knife, serrated knife, to a electric powered, saw. But recently I bought a small axe and I've just been chopping it with an axe. I mean, they're so tough that with an axe, I can actually just slice it and I get really thin slices. And that works really well for not just cloning, but also for making really nice tea because I can powder it up easily. Once they get this hard though, what I just said right now is with fresh mushrooms. With something like this that's fresh, an axe will cut through it really nicely. A knife, you're going to work really hard with it. But once it gets this hard, man, it's really hard. It's really difficult to do it. So whatever you can get your hands on, bandsaw. Really, the recommendation is cut it fresh and then dry it, and then that'll be good enough to preserve it, or try to clone it right away.
Yeah, I've used power tools before to cut them, and it works pretty well.
Yeah.
But yeah, at that point, I don't even know. I've done it when it was fresh. So.
Exactly.
Yeah. So, here's a really good question. What happens to heavy metals after fungi assimilate them? Aren't they still in the environment?
Yeah, we didn't talk too much about heavy metals today. It's pretty complex stuff, but yeah, fungi can break down things like hydrocarbons and agrochemicals, down into some other less harmful byproducts. But heavy metals can't be reduced further than their elemental form. What can happen is that they can be oxidized or reduced and change the bioavailability of them, right? So even though they're still present in the environment, they're locked away, immobilized, so that plants can't take them up, and then something else can come and eat that plant, and then goes down the food chain, or something else happens to them. The fungi are able to simply trap them, immobilize them, either chemically or mechanically if we grow mycelium on contaminated substrate, turn it into a brick, dry that brick out, and now it's locked away, right? We can use it for construction, for example, right? Those heavy metals aren't going to go anywhere, right? So that's the basic stuff with heavy metals. Once again, it's pretty complicated. I focus mostly on organic contaminants, but I'm happy to refer some of y'all to more heavy metal stuff if y'all need.
And I assume that micromediation can't be used in the ocean.
Well, do we want to? Do we want to use fungi in the ocean? Do we want to ignore the natural requirements for fungi, right? Which is that they breathe oxygen, they release CO2, they need a source of food, they need room to breathe, they need room to expand. Can we achieve those parameters in the ocean? That's the first thing. Do you want to do that? If so, there's so many different ways to do it in the ocean, but it's going to involve so much more work, right? My approach here, my mindset here is, let's work with what fungi are already good at. Let's instead of putting wood decomposers, which there's no trees living in the ocean and falling and then, so that doesn't make sense. But what we can do is bring the contamination from the ocean over to land, right? Maybe create a system in which we collect, we absorb oil from an oil spill in the ocean with booms, is what they call them, right? Either made out of straw or something else that will absorb it, but that can also serve as a source of food for wood decomposers on land to break down. And now we've made our life easier, and we're increasing the chances for our wood decomposers to do that work. Now, obviously, there's a whole nother there are other categories of fungi that I didn't mention in this talk, that may be able to do the work on site in the ocean. There's a whole nother field that's called marine fungi, or a whole other category that are marine fungi. And I've slowly been reading about them. So maybe they would be able to do the work in the ocean. But with the wood decomposers, you don't want to do that in the ocean, right? They're also not salt tolerant. We could increase their tolerance to salt in the lab. But once again, it's about how far do we want to push the limits of fungi and what they do best, right? And I'll just wrap it up with that.
Word. Someone said in the Zoom chat that people used human hair waste to absorb oil in San Francisco Bay, and then collected that and composted it with oysters to break it down. But when you were
Yeah,
talking about the enzymes earlier, it kind of got me wondering, like, couldn't you just, like, in a biodigester, like, collect a bunch of these enzymes and then apply that just through, like, because, the oil floats on top of the water. Couldn't you just, like, break it down that way? Or is that still kind of too hypothetical at this point, just
it,
because we don't know enough about the types of enzymes that they produce and the efficacy and, like, kind of that stuff that you were talking about?
yeah, yeah. I mean, you're totally spot on. It's definitely something that I want that it's within the line of work that I do. Is instead of going back to the large scale examples that I gave y'all, where we introduced massive amounts of mycelium to the spill site, can we introduce a sample of the contamination over to our bioreactor, or large scale liquid culture reactor, or very simply, in our large scale mycelium cultivation techniques, and allow the mycelium to react to that contaminant, producing this particular cocktail, biochemical cocktail, and then take the biochemical cocktail, dilute it and expand it, and take that over to the contaminant. That's definitely one of the things that I'm working on. It hasn't really been tested. It definitely hasn't been tested in the ocean. Um, but if you do want to see some examples of that, I've got some papers that have tried it, not so successful, because that chemical cocktail, it's still kind of like up in the air. How do we preserve it? Do we need some sort of medium to carry it in? One of the things that I'm testing out right now is using biochar. Right, I make biochar, and then I inoculate it with the raw enzyme exudate, or the biochemical cocktail, to then introduce it to these contaminated sites. That's one approach. The ideal would be to just have like a tank that just sprays this biochemical cocktail, and it's a liquid application. That would be ideal, and then maybe we can do that in the ocean. But at this point, like you said, it's just speculation. But it is backed by everything that I said, which is like natural ability, natural processes of fungi, and it makes sense to do it. It's just that we're not really good at it yet.
Yeah, so, like, kind of at the point of learning the best ways to get the enzymes out there, whereas, like, the fungi obviously have had billions of years to evolve that process of making it stable and all that stuff. So,
Exactly.
yeah.
Exactly.
Um, so, any more questions out there? I know we're kind of close to the end here. Daniel's probably got all sorts of fun stuff to do down there in the Yucatan. So we won't want to keep him much longer. So if anybody has any more questions, feel free to chat. YouTube, Facebook. There's actually someone out there on the Twitter watching. Yeah.
What's
so
up, Twitter?
You got a question? Shout it out. No dumb questions. Let's see. Was that a Gen 5 sample from the Yucatan?
Which one? A Gen 5?
Yeah, I don't understand.
Oh, yeah, maybe from the plates. Are you talking about the generations? Yeah.
That's it, G5. I can read your handwriting.
Oh, GS. It's a Ganoderma Cecile. Yeah.
Nice.
Is the
GS.
Is
There
that
you
where
go.
you found it?
Yeah, I found it down here in the Yucatan.
Oh, I have a question. And
Sure.
this kind of relates to some of the more like moving into the future. But like, are you doing any DNA analysis with this and like kind of trying to tie that back into it? Because that obviously could play into like some like CRISPR stuff in the future too.
Yeah.
It's like, oh well, we need a new enzyme. Obviously, like I'm on board with like stopping this kind of pollution and like, but you know, we do have a lot of it that exists already. But yeah, so like, how are you doing any DNA research with this, or is that something you hope to expand on or get someone else interested in?
Yeah, I'll be up in Chicago working in a lab to do more of this work. But part of the conversation and part of the scope of that work is to explore these topics of gene expressions, right? Because ultimately what we want to, or maybe not what we want to do, but what we're able to do with some of these techniques, like, now that DNA sequencing is getting really cheap, relatively cheap, and things like CRISPR is coming about. Can we actually find the genes that are being expressed that are responsible for the production of these enzymes and other exudates? And could we somehow transfer this code or these genes over to something that's simpler, that's easier to grow? Because fungi, although, I did emphasize the fact that they're easy to grow, something like yeast or E. coli is even easier, right? So if we can like somehow modify yeast to simply express these genes to produce this chemical cocktail, that could speed up the process of getting to the liquid application that I mentioned. But I simply don't know enough. I just know that I'm going to Chicago at the end of the month for a couple of weeks to explore these topics. And so, but I am in conversations with people like Alan Rockefeller and Craig up in New York, who have way more experience and are way more knowledgeable, and they're guiding me. They're guiding me to do this kind of work as well.
Very cool. Yeah, this seems like definitely a place that needs so much more, an area of study that needs so much more funding. So get the feds to give you all a bunch of money. That's what should happen.
The feds.
When are you visiting Austin again?
I don't know. Whenever you guys invite me to come up.
Yeah, there you go. Are you planning a public mushroom trip to Mexico or in Mexico soon?
Yeah.
Because I remember you had one, and then COVID canceled that.
Yeah, that was a bummer. I'm shooting for November because that's the main season here. October to December is when it's not so hot. There are other places in Mexico where the season is just starting now, and I'm going to go visit those places too. July, August. There's places in like Oaxaca where August, they call it Ongosto. Like it's just dedicated to mushrooms because it's the prime season. But here in the peninsula, in the Yucatan Peninsula, it's from October to December, just 2-3 months where it's really nice. And so I'm shooting for late October, early November for a trip. And so the ideal scenario, and that's something that I'm still trying to figure out, is take 2 weeks, for example. 1 week in which I'll expose you to many different things, from micromediation research to cultivation, medicine making, right? Extracts, spagarics, things like that. And then going out to visit some other projects. Then once you get exposure to the different things, the second week you get to choose. What do you want to focus on? Because at the end of the 2 weeks, what I would want you to do is to have something to walk away with, right? So if you want to explore the medicine route, right? You want to make some tinctures. Well, at the end of that, I want you to have a nice little blend to take home so that you can take that and start a business with it, right? Or if you're interested in the micromediation stuff, you'd be able to conduct some research, take home some practical skills, but also walk away with a nice little set of data that you can build off of. And then from there, all the different tracks, anything from identification. I'm bringing in some actual mycologists that know what they're talking about in the field to help people, understand mushrooms in the wild or identify mushrooms in the wild. And that's the general idea. I've got to figure out the logistics, but I'm shooting for November. So if anybody's interested in doing this kind of stuff, spending some time with me down here, I'm preparing a lot of really cool things. So reach out, and you'll probably hear about this at the end of the summer.
Very cool. Well, it seems like there's no more questions. You have someone in the chat saying they took your myco class at the community garden a while back and they're still growing mushrooms, living the dream. So I thought you'd like to know that too.
Yes, thank you.
But if we don't have any more questions, I think we'll wrap it up for the evening. So yeah, if you liked our lecture tonight and would like to help support more of them, you can obviously like and subscribe to like the video, subscribe to our YouTube channel, but you can also head on over to the centraltexasmycology.org and become a member. You get access to a Discord community where we talk about mushrooms and all this, different kinds of projects going on, talk about mushrooms that we find, that kind of stuff. You also get discounts to any classes, members only forays where we go out and into the woods and hunt for mushrooms. So that's some of the benefits to membership. You can also make donations there, sign up for our myco block distribution to get used mycelium blocks from some of the local mushroom farms, keep them out of the landfills. That's centraltexasmycology.org. Thanks, Daniel, for talking with us tonight. And
Philip, one more thing.
Oh, yeah,
Remember,
go for
it. So I also wanted to just jump on what you said, which is that, encourage people to become a member. And if you are limited in resources for whatever reason, I'm sponsoring some folks that can't pay their way into the membership through this talk, right? I'm not charging anything for this talk. I want this to go to people that can't afford a membership. So if you are intrigued by what Philip said and everything that CTMS has done, just let them know that you're unavailable or unable to make that membership fee, and it'll be taken care of. Okay.
Awesome. Thanks, Daniel. Yeah, shoot us an email at info@centraltexasmycology.org with I don't know, Daniel membership or something on the tagline. Figure out some special way to notify us that this is what you're emailing about.
Just say, I'm broke, but I wanna join.
Yeah. Financial assistance membership, something like that. And yeah, we'll totally help anybody out, really. But we really appreciate, Daniel, for you donating your fee for that tonight. Thank you.
Awesome.
And with that, I think we will sign off and say, and good night, everybody.